from MPN Advocacy and Education International, the CEO and founder. We have many of you joining us this morning, and I'm so pleased to be able to do this. It's our first time out, so bear with us. There may be some delays, there may be some questions that we miss, but we will certainly try to answer any questions you may have. I will be getting those questions as well as Kathleen, and we will field those and ask the appropriate question to the appropriate physician that speaks this morning. We also want to let you know that we are here. We've been open, we're working. We are trying, I hope you're receiving our weekly updates. Kathleen has put those together beautifully to try to keep you informed and to let you know what's happening with COVID-19. Our speakers today are dear heart, Dr. Ruben Meza, who is our scientific advisor and speaks more often at our in-person events, is with us this morning, and he will be discussing MPNs and COVID-19. And then we'll hear from Dr. Aaron Gerds. Before that, I would like to extend a very deep appreciation and thanks to our sponsors and industry partners. And I would like them to give you a 3-minute update on what's happening in their arena and with their drugs right now. And then we'll hear from Dr. Meza. Thank you. First, I'd like to introduce Laura Eichhorn from Imago Biosciences. Laura?
Good morning, everyone. Thank you, Anne, Kathleen, and the entire MPN advocacy team for your tireless dedication to the MPN patient community. I've been working with Anne for the past few years. I'm inspired by her commitment and impressed by her passion to provide support and information to each of you. Whether you're a newly diagnosed patient, a family member, or support person, I would encourage you to take advantage of the wonderful resources that the MPN Advocacy and Education International has to offer. The physicians who are presenting today, Dr. Gerds and Dr. Meza, are globally recognized leaders in this field of research, and we're honored to be a part of this presentation today. Imago Biosciences is a private venture-backed company founded in 2012 based in South San Francisco, California. We're entirely focused on the treatment of MPNs. Those diseases pose great challenges to physicians and patients as well as their families. Our approach to these diseases is through the study of epigenetics, the science of gene regulations that does not involve changing genetic code. Bomedemstat drug, which is also known as IMG 729, was discovered by Imago in 2015, and it's an oral medication taken once daily. In 2016, bomedemstat was first taken by patients with leukemia to study the safety and to understand how the drug is broken down and eliminated by the body. Bomedemstat inhibits an enzyme that regulates the maturation of blood cells in bone marrow. It is the goal of the therapy in myelofibrosis to improve symptoms, reduce spleen size, and reduce the number of malignant cells. For those with essential thrombocythemia, the goals are similar, including reducing the platelet count to normal. Bomedemstat was designated by the FDA for fast-track approval for the treatment of both myelofibrosis and essential thrombocythemia. For the treatment of myelofibrosis, Imago is conducting a small phase 2A clinical trial in 18 patients to understand the safety and efficacy and dosing of bomedemstat taken daily for 12 weeks. There were no safety signals in that portion. Most patients experienced improvement in their symptoms and shrinkage of their spleens. These results encouraged us to move forward. Therefore, Imago is now conducting the second portion of the phase 2 trial to further study the safety and efficacy of bomedemstat taken daily for 24 weeks. The starting dose is higher with the belief that this will lead to more rapid improvement in symptoms and reduction in spleen size. Dr. Kristen Pettit of the University of Michigan and a clinical investigator in this study will be presenting the data at the upcoming virtual European Hematology Association meeting this June. An abstract is now accessible online that highlights the current data demonstrating a reduction in spleen volume and an improvement in symptom scores. Most patients also had a stable or improved bone marrow fibrosis score and hemoglobin. Overall, preliminary data in this ongoing study show that bomedemstat is generally well tolerated in a diverse population of patients with advanced myelofibrosis with limited therapeutic options. The study has been expanding and is currently enrolling at sites in the US and in Europe. However, enrollment in this study remains open, although many centers have reduced access due to the current local restrictions as well as a result of the COVID-19 pandemic. The safety and efficacy of bomedemstat in the myelofibrosis study encourages us to expand our clinical research in other myeloid diseases. We will soon begin a study of bomedemstat for the treatment of essential thrombocythemia. Bomedemstat will also be tested for the treatment of polycythemia vera at the University of Miami. Imago will also begin this year a study of bomedemstat added with ruxolitinib for the treatment of myelofibrosis to determine if there is any additional clinical benefit of the two agents together. We're currently conducting research in the use of bomedemstat and related compounds for the treatment of leukemia, sickle cell disease, and solid tumors. Thank you all for joining the meeting today, and again, thank you, Anne.
Thank you, Laura, very much. And we really appreciate it. Now we'll hear from Mark Kowalski, who is the Chief Medical Officer at Sierra Oncology. Mark?
So Sierra is developing the therapeutic product momelotinib for the treatment of intermediate and high-risk patients with myelofibrosis. Just very quickly, some background on momelotinib. Momelotinib is a JAK inhibitor that also inhibits the signaling pathway called the ACVR1 gene pathway. The ACVR1 pathway contributes to the anemia, which so often occurs in myelofibrosis. So therefore, and importantly, momelotinib is different from the other JAK inhibitors because it often provides a beneficial effect for the anemia in myelofibrosis in contrast to the worsening of anemia seen with other JAK inhibitors. So by targeting both JAK1, JAK2, and ACVR1, momelotinib is able to inhibit all three key drivers of the disease, and by doing so, momelotinib addresses the three main features of myelofibrosis, which you know are anemia, constitutional symptoms, and splenomegaly. So momelotinib's benefit on myelofibrosis has been extensively studied already in over 820 myelofibrosis patients having been treated in multiple clinical trials. So moving to the MOMENTUM trial, Sierra Oncology is now actively enrolling a phase 3 trial globally called the MOMENTUM trial. We're open for business despite the COVID. We are enrolling patients now. So the MOMENTUM trial is recruiting myelofibrosis patients who are symptomatic and anemic and who have been previously treated with an approved JAK inhibitor. So currently, these would be ruxolitinib or Jakafi or fedratinib and Repic. So the study aims to determine the activity of momelotinib on the three key disease manifestations, as I mentioned, symptoms, anemia, and splenomegaly. The study is going to compare momelotinib to danazol, a treatment that has been used for many years for the management of anemia of myelofibrosis. The study will randomly assign patients to one of these two treatments. So two patients will be randomly assigned to receive momelotinib for every one patient assigned by essentially a flip of a coin to receive danazol. The trial is blinded. That means that neither the patient nor the physician will know which treatment the person is receiving, but all patients will eventually be able to receive momelotinib. Even the third of the enrolled patients, which were initially assigned to receive danazol, will be able to receive momelotinib treatment after 24 weeks, if not sooner. That is, there's provisions for early crossover to momelotinib should the need arise. So many sites are already open in the U.S., including the Cleveland Clinic and Gabrail Cancer Center in Ohio, Washington University School of Medicine in St. Louis, the American Institute of Research in Whittier, California, and also in California, the Irvine Center for Clinical Research. We're open at the University of Texas in San Antonio, and Northwest Oncology and Hematology in Rolling Meadow, Illinois. MOMENTUM is truly a global trial with sites also in Canada, Australia, Asia, UK, EU, and Israel, in addition to the US sites, and we're opening more US sites. So you can learn more about MOMENTUM by visiting the website momentumtrial, that's one word, .com, or contact us at info@momentumtrial.com. And I thank you very much for listening to me today.
Thank you so much, Mark. That was great. As those of you join us, we're hearing from our biopharma industry partners and sponsors for this program. As you can see on the screen, our speakers today are Dr. Ruben Meza and Dr. Aaron Gerds. And so next we'd like to hear from Meredith Manning from Pharma Sencha.
Hi, good morning. Thank you for inviting Pharma Sencha to speak with you today. I'm newer to the company. I recently joined in February of this year, but I know that Pharma Sencha has had a really successful, long, fruitful relationship with the MPN advocacy organizations across the US as well as globally. So for those who are not familiar with Pharma Sencha, we are a global pharmaceutical company. We have organizations not only in the United States, where we set up our US subsidiary in 2017, but we have our headquarters in Taiwan, and we also have additional subsidiaries set up in Japan, South Korea, China, as well as a subsidiary in Hong Kong. And we'll be looking at other countries as well as we move forward. I'm excited to join as the general manager of the US subsidiary, working with Craig Zimmerman, who many of you know, who's been speaking to the organization. And we're excited. We have a couple updates with regard to ropeg interferon alpha 2B, which is our product. We developed that product, or founded that product, in 2008. So we've been working for a long time to bring ropeg interferon alpha 2B to the marketplace. Our recent update is that in Q1 of this year, we submitted to the FDA our BLA, and hopefully we look forward to providing updates to the community in the near term as to when we're expecting to go before the FDA and hopefully receive approval. So we'll be very open with our information and share that with you all as that comes along. But we're excited to bring ropeg interferon alpha 2B to the marketplace in polycythemia vera. The other update that we have is we're looking at starting a clinical trial in ET, and we will also share additional information. As of right now, we are working with approximately 10 sites, and we hope to open that trial in probably at the end of Q3 of this year, 2020. So those are our quick updates with regard to Pharma Sencha, and excited to be part of this community. In the past, I've worked over the years in both hepatitis C as well as hemophilia, and so I'm excited to learn more about this community and be a part of this community, as Pharma Sencha has been dedicated for over 20 years to bringing ropeg interferon to market. So thank you.
Thank you so much. I truly appreciate that. Our next biopharma person, who is our industry partner, is Sarah Buckley, and she is with CTI Biopharma.
My name is Sarah Buckley. I'm a medical director at CTI Biopharma, and we are developing pacritinib, which is a JAK2 and IRAK1 inhibitor for patients with myelofibrosis who have severe thrombocytopenia. And I'd like to take a few minutes to tell you about pacritinib and about the phase 3 Pacifica trial, which is currently open to enrollment. Now, pacritinib has been studied in over 1,000 patients across 15 clinical trials, including 2 phase 3 studies in patients with myelofibrosis. And those are the PERSIST-1 and PERSIST-2 studies. And in both of these studies, pacritinib was more effective than best available therapy at reducing spleen volume. Now, both PERSIST studies included patients with severe thrombocytopenia, which we define as platelet counts less than 50,000. And this is an important point because patients with MF and severe thrombocytopenia often have greater symptom burden and disease burden than patients with higher platelet counts. And we also know that treatment options for those patients with severe thrombocytopenia are limited. Clinical trials often exclude those kind of patients. So we recognize that these patients face an unmet need for effective and safe therapies. At ASH this last year, CTI presented results from our phase 2 dose-finding study. This was called the PAC-203 study. And PAC-203 enrolled patients who did not tolerate ruxolitinib or who failed to benefit from ruxolitinib. And it included patients with severe thrombocytopenia. Almost half of the patients who enrolled had platelet counts of under 50,000. This protocol also included enhanced safety measures to help us monitor for side effects. From this PAC-203 study, we were able to select a dose that demonstrated clinical activity and had an acceptable safety profile, even in patients with severe thrombocytopenia. And so this is the dose that we've brought forward into the Pacifica study. Now, Pacifica enrolls patients with MS and platelet counts less than 50,000. Pacifica is open to patients who have either had no prior exposure to JAK inhibitors or who have had a limited prior exposure. So patients with up to 6 months of prior exposure may be eligible. Pacifica enrolls patients who have splenomegaly and symptoms related to myelofibrosis. And patients would be randomized 2-to-1 to pacritinib versus physician's choice therapy, which includes a choice between ruxolitinib, danazol, hydroxyurea, or corticosteroids. And so that means that for every patient randomized to the control arm, 2 patients would be randomized to the pacritinib arm. And enrollment is currently underway, even with COVID. The study is open in the US. We're in the process of opening around 100 sites in the US and worldwide. So you can find more information about this at clinicaltrials.gov or at the CTI website, which is ctibiopharma.com. Thank you very much.
Thank you so much. I truly appreciated that. And now we will hear from Leanne Holes from AbbVie, who is also, they are a new sponsor in the last year, and we truly appreciate them. And Leanne, if you're there, please give your update. Thank you.
My name is Leanne Holes, and I am a scientific director at AbbVie. AbbVie is a global pharmaceutical company dedicated to bringing new therapies to patients through a multitude of different diseases. We are currently developing navitoclax in myelofibrosis, and navitoclax is an anti-apoptotic small molecule that binds BCL-XL and BCL2, which can free apoptotic, pro-apoptotic proteins to initiate programmed cell death. We've done quite a bit of preclinical work that provides rationale for the role of BCL-XL in myelofibrosis, indicating that it has the potential to prevent or reverse bone marrow fibrosis, as well as the combination of BCL-XL inhibitors with JAK2 inhibitors may work synergistically, and that the inhibition with BCL-XL can overcome resistance to JAK2 inhibitors. We have seen encouraging interim efficacy data, which was presented at ASH from our ongoing phase 2 clinical trial. We initially enrolled 34 patients, and we have recently expanded this study to enroll an additional 130 patients, both in patients who have received prior treatment with ruxolitinib or another JAK2 inhibitor, as well as patients who have not received treatment with a prior JAK2 inhibitor. Patients will receive either the combination of navitoclax plus ruxolitinib or navitoclax as a monotherapy. The primary endpoint is to evaluate the effect of treatment on spleen volume as well as symptom control. Additionally, we have, for this study, we have about 40 sites globally. We are enrolling despite COVID-19 right now, and we are currently activating more sites as we're doing this expansion. We have a number of sites here in the US, including about 20 to 25 sites at most of the centers across the country. So you can find more information regarding this clinical trial at clinicaltrials.gov or at abbvie.com. We are also planning to initiate 2 phase 3 studies in the frontline population who have not received prior treatment with a JAK2 inhibitor, as well as patients who have received prior treatment with a JAK2 inhibitor later this year. Thank you for your time.
Very interesting. And as patients in this MPN space, I'm telling you, there's so much going on. It's just incredible. I'd like to thank all of our sponsors, including Insight Corporation. Kristen, I know you're on, but will not be presenting anything here, but I know a lot of work is being done with Jakafi and COVID, and we have had some other MPN experts talk about that in previous videos. So at this time, I would like to introduce our first speaker, a friend to all of us and a wonderful man for MPN Advocacy and Education International. He's been our scientific advisor and pal and frequent speaker at our in-person events, which we hope one day will resume. Not anytime soon, however. And so without further ado, please welcome our dear friend, Dr. Ruben Meza.
Hello. Well, it's a tremendous pleasure to be here today. Greetings from San Antonio, Texas. Dearest thanks to Anne and the organizers for putting this together. You know, we know that education as it relates to MPNs is always important, but I think even more so during this time where not only are you worried about your MPN, but obviously you are very much in the middle of this pandemic with the rest of society, but with some special potential considerations and things of that nature. So both Dr. Gerds and I thought that the focus this morning really should be related to how COVID is impacting you as MPN patients. We'll certainly be touching base on the new therapies in development, but without question, there are many issues not only related to MPN therapies, but really related to COVID and yourselves that are crucial during this time. So I'm going to share some discussion that I've held in a variety of venues here. So first, my disclosures, I pretty much have a relationship with everyone in one form or the other in this field and appreciate those relationships. So I'm going to be talking on COVID and MPNs, and maybe start off with a high level comment that, you know, you hear about all these wonderful therapies in development. And Dr. Gerds will be talking a little bit later about clinical trials. And indeed, continuing the clinical trial process is really essential. These new therapies in development are important, and this is a crucial time for these trials to be able to move forward so that these therapies can be an option for MPN patients. I think now it is safe for people to be able to go in to see their healthcare providers, to enroll on studies and be treated with these therapies for several reasons. The beginning of this time, everyone was scrambling around to try to figure out how to do this in a safe way. Indeed, at our cancer center, we too have evolved, but now have a process where we believe it is very safe. People are screened. People are screened before they enter in the building, even before appointments are made. There's universal masking, deep cleaning of surfaces, social distancing, on and on. So currently, it is not a time that you should feel the need to have to curtail whatever healthcare you truly need, whether that be related to your MPN or to other issues. Now, as it relates to MPNs, there has been a very active discussion really from the very beginning of this, going back to March. And the American Society of Hematology pulled together several of us involved with MPNs, particularly many colleagues in Italy and Europe that were somewhat preceded us into this crisis. And we developed a set of discussion points, realizing that we don't have all of the answers, but discussion points as best we can to guide treating hematologists as it relates to MPNs and COVID. And you are able to find this on the ASH website, hematology.org. It's written for physicians, but certainly it is public knowledge, and realizing that in terms of data for MPNs is only something now that we are learning. And certainly this was a team effort, as you see, and recognize many names on this list. Now, COVID, I'll start with a few slides that are shared with me by our colleagues in infectious disease here at UT Health San Antonio. And we've been really grateful for their leadership. These were individuals that were engaged with the SARS crisis in Toronto in 2003. So fortunately, we had some real seasoned experts that helped guide us in this process very early on. Now, just a bit of a highlight. You hear so much information, and some of this may be duplicative for some folks, but I think it's helpful to at least have kind of a shared standing. So realize we only learned about this in December of 2019. So you see, really in a 6-month period, well, almost 3 of which have been under lockdown, how much has evolved, learned, and how much impact that it's had. As it's well known now, it started in Wuhan, China, potentially associated with this seafood wholesale market. Now, coronavirus is a description for a broader term for a group of viruses. It's not just a single virus. That's why sometimes you hear it referred to as the novel coronavirus. There have been 4 variants of the coronavirus circulating for many years, including the common cold. SARS, the 2003 pandemic in China and Toronto, was another coronavirus. In some ways, this is a mutation of that virus. MERS was another coronavirus that had issues again with another animal, camels. And the specific virus name in 2019 is SARS-CoV-2 2019. So COVID-19 is the name for kind of the illness that transpires. The name of the virus is SARS-CoV-2 2019. So that might help clear up some of the confusion that sometimes you get as people will refer to it one or the other. Now, as we look at these different coronaviruses, and in this column, this is the virus that we're dealing with now. Again, there was thoughts that the animal to human jump point were bats or these small animals called pangolins, an incubation period of 2 to 14 days. That's why you hear so much in terms of different sequences during this time of 14-day windows. The infectivity rate makes it quite infective at this potentially 2 to 3 rate. The rate of asymptomatic transmission, the potential for healthcare transmission, although with proper PPE, masks, and other things, fortunately, that has been relatively low. And we don't still yet know the fatality rate, although regardless of what that number will be, clearly it has been too high in terms of the unbelievable toll that it has had. Now, in terms of transmission, these are the number of average secondary cases. And again, it's estimated it's probably 3 or more. It's difficult to know because of the limitations in testing how many people have actually been affected in an asymptomatic way and recovered. Seasonal flu, that's about 1.5. The pandemic of 1918 that we've heard so much at was at about 2. It is somewhat comparable perhaps to measles, which is very contagious, although fortunately contained by vaccination. Now, as we look at other pandemics, if we look back to the one that's discussed so much, the 1918 pandemic, again, there were probably over 500 million deaths, US deaths of almost 700,000, global deaths, or infections, I should say, of greater than 500 million, global deaths of 30 to 40 million, and then subsequent pandemics. Your typical seasonal flu, about 22 million infections, about 22,000 people died, a case fatality rate of under 1%. This showing a bit of kind of the incubation period. That's why you hear so much about people isolating for 14 days or waiting 14 days to see if there's going to be a surge. That's why so much of this has been in kind of 2-week increments. Now, in terms of age distribution, again, the majority is in the people of average age. This is not too dissimilar from the age distribution of the population. Now, the mortality is higher in this older group, but it does not protect us for individuals more in kind of the middle of life. And sadly, we all know examples, either personal or in public knowledge, of people that were younger and still had a significant consequence from the virus. Now, in terms of severity, again, the majority of people with this virus, fortunately, will have a mild illness. And when questioned, many of the asymptomatic folks probably had some mild symptoms at least, body aches, even if they did not have fever or things of that nature. With a smaller percentage, but this is higher than most similar illnesses, having a severe or critical phase. Now, in terms of fatality, again, very heavily skewed toward older populations, having a much higher rate of severe illness. Now, why the severity? Well, we're only starting to learn all of the different consequences of becoming infected with this virus. But what happens is that in a minority, there is a pneumonia meaning infection in the lung tissue itself that has a lot of inflammation. And with that inflammation, it makes it difficult for the lungs to transport oxygen to your blood, their main function, and it makes them stiff. That is why individuals have needed mechanical ventilation to help to support or try to overcome that difficulty during the critical period of time. That's why there was so much focus on number of ventilators, respiratory support, ICU beds, etc. Fortunately, that has been the minority of individuals, but obviously at a much higher rate than would be normal, and not without consequence. People that end up on ventilators, not all of them clearly survive that difficult process, and they can be left with long-term medical problems. So it is something that we clearly want to try to avoid if at all possible. Now, in terms of testing has evolved very quickly, where many centers develop their own tests, frequently with kind of FDA parameters. We have an FDA-approved test that we developed here at UT Health San Antonio, but that's not unique. Most centers develop their own test because the other process was slow in rolling out. Now, at the beginning, testing for this has been somewhat uncomfortable. As you could see, the swab goes way back into the back of the nose. There's now all sorts of research being done trying to test, not going nearly that far back, some with some good accuracy to make it simpler. And both access to the reagents for the test has been a challenge, and even the swabs themselves, because of their length and very specific nature. All of that, I think, is largely worked out. Now testing is being done at a much more significant rate, but that's part of the reasons why it has been a slow process. Now, in terms of therapy, we, in short, don't have an easy cure for this virus. If we did, this last 2 months would have looked very different. Obviously, for the majority of people that are asymptomatic, there's no therapy at all. Individuals that are hospitalized and sicker, clearly we are trying to support them. They need oxygen, and now there is a concern that there is a higher rate of blood clots that can form in individuals with this infection, sometimes delayed, sometimes in those that were asymptomatic. So there has been use of anticoagulants in a preventative fashion. We'll get back to in a moment what that means for patients with MPNs. The hydroxychloroquine story is evolving. I won't get into the politics that's surrounded this agent. So far, it had looked potentially promising. Unfortunately, I think due to, in particular, toxicities from the agent, as well as lack of clarity whether it was truly effective, I think it's largely going to be falling into disfavor, in that the large clinical trials have been stopped due to lack of effectiveness as well as side effects. Now, remdesivir is an antiviral therapy. This is the one that was approved in a rapid fashion. It had been used, I believe, for Ebola. We did have a clinical trial of it here at UT Health San Antonio. This is a powerful intravenous antiviral therapy given for individuals who are not only hospitalized, but also very sick. So this is not something people are going to receive, unless if they are very ill. A study that has been led by my prior institution, the Mayo Clinic, they've been using convalescent plasma. So people who had the virus, have recovered, have antibodies in their blood that can fight the virus, they were able to collect blood. They are able to filter out the plasma where these antibodies will be held, and then give this plasma to individuals affected with the disease. There are clinical trials ongoing with this, but this has been somewhat helpful as well. There is clearly research. There are multiple clinical trials. And again, the JAK inhibitors that you have come to hear so much about, whether it be ruxolitinib, which had helped to decrease the inflammation in those patients, whether it pacritinib, momelotinib, all may potentially play a role. not related to their functions against MPN. This is related to, again, their functions against inflammation, JAK1 and JAK2, decreased inflammation, decreased severity of the pneumonia. There has been research across the world at a scale that has never been really seen before. This also includes research in terms of development of new personal protective equipment, tons of research regarding multiple different sorts of vaccines that are being developed in parallel, new therapies, better understanding the immune system, on and on. There's also research understanding impact for cancer patients. There's research ongoing that Dr. Girth, myself, Dr. Mascarenhas, and others are putting together regarding even how we help care for MPN patients during this time. So lots of research for us to learn more. Now, when are people released from isolation? People who've had symptoms on home isolation may discontinue after at least 3 days since recovery, a resolution of fever without the use of fever-reducing medications, and improvement in symptoms, and at least 7 days since the symptoms first appeared. And again, I would advise all to be using, if you're in a situation with other individuals, to use universal masking during this time. So this does not mean if you're running on a country road by yourself that you need a mask. Obviously, the masks are really if you're around other individuals. But if you're in stores, grocery stores, public transportation, on and on, if you're around other people, masks really do help. Now, all of this has been to achieve what has been called flattening the curve. So what do they mean by that? Curve relates to the number of cases and the number of cases at one time. The entire effort to flatten the curve was to really be able to preserve enough healthcare capacity in the system to be able to care for the number of people who got very ill. That is where certain areas were under tremendous strain, as you saw very much on the news, areas in New York and the Northeast, in particular in the US, in Italy, in Spain, in China. Other areas, the healthcare system had the capacity. Now, even with the curve flattened, that did not mean that people were not dying from the infection. Clearly, we've lost now about 100,000 Americans, many in states where the curve was flattened. So it doesn't decrease the severity of the illness. It just means that there is capacity within the system. You know, I've seen some of the discussion on social media. Flattening the curve only means that there is now room in hospitals if you become ill. It does not mean that you're protected against the infection. So how do you prevent the spread of this infection? Fundamentally, the main way that you get this infection is that it comes in somewhere through your eyes, nose, or mouth. So it does not get in through the skin. So that's why the things, one, trying to avoid other people the best you can within reason is helpful. Again, the virus has not left our society. So again, be mindful of this to the best degree that you can. Clearly, this is where washing the hands is essential. People say, well, why don't I just wear gloves? It's actually not about your hands. It's about your face. So if you wear gloves, you still would have to clean the gloves, because if you get it on your hands and you touch your face, whether you're wearing gloves or not, you still can become infected. So to some degree, we've really not been encouraging gloves because they don't really accomplish anything in this process. And actually, they probably tend to make people feel artificially protected where they would not be. As we're really trying to avoid touching your face, particularly in public, is so helpful, because that is the primary way that people are getting the virus. Also, it really stays on surfaces. You know, so when I'm out at the grocery store, again, I have my mask on, I wash my hands, you know, I have the cart that's been cleaned. You know, I try avoid touching only those things that I need to touch, but I really try to avoid touching my face at all in the grocery store. Get back out to the car where I have my alcohol stuff and totally douse my hands before taking off my mask. Now, other things you can do, people ask about nutrition. there is no magic bullet, but clearly eating well is helpful. Probably a multivitamin is helpful in this period as well. We have found that there's not an insignificant number of people in the U.S. who are obese. They have plenty of calories, but they don't have a lot of the micronutrients and minerals that are helpful in the immune response. That's where vitamins can be helpful. So a general healthy diet is helpful during this period of time, as it is at all times. Adequate sleep is important. This is helpful in health and disease, and it's certainly helpful for your immune system to function the best way that it can. Also important is to manage your stress. I would limit the amount of time you're watching the news on a daily basis, in that, again, it really is, it's a difficult set of news, hopefully slowly improving, but it creates a tremendous distress. Clearly, we're coming into summer. There is nothing about fresh air that is going to give you the coronavirus. Go outside, have activity, meditation, certainly connect with people, just not within 6 to 10 feet. That can be through social media, that can be through FaceTime, that can even be in other venues outside, but at greater distance. Now, how does this relate to MPN patients specifically? So this has been kind of a broad overview as it relates to COVID. So how do we link it to MPNs? So first, in terms of data specific to MPNs, we are only starting to learn more. I can share, we've been in close contact with our colleagues from Wuhan, China. So they have the longest experience. And again, they did not find a specific signal as it related to MPN patients and COVID. I think we will learn much more through some ongoing studies, including in Italy, where Dr. Tiziano Barbui in the European Leukemia Net is gathering data on MPN patients in Italy and in Northern Europe, where the rates of infection were very high, and many of us from American centers contributing. So the first question is, are MPN patients at higher risk? And as I think at risk as it relates to COVID, is it an increased risk of becoming infected, or is it an increased risk of complications if you have the COVID infection? So let's try to tackle the first one. Are you at a higher risk of developing the infection? I don't think we know, and we do not have definitive data to answer that. I think we have at least a concern that some patients with MPNs, if they're more debilitated, if they have more advanced myelofibrosis, potentially if they're on a JAK inhibitor, may have a slightly higher rate of acquiring the infection. We don't know that for certain, but we are advising those individuals to be more cautious. We don't necessarily believe that ET and PV patients are necessarily at a higher risk of acquiring the infection, but we are advising everyone to be cautious. We do not have any definitive proof that therapies like aspirin, phlebotomy, hydroxyurea, or interferons necessarily increase the risk of developing the infection. Now, interferons have some antiviral properties, so it's possible they may have some protective effect against getting the virus. Now, we don't know that for certain. We don't want anyone to count on that, but that is possible. Additionally, people who are on JAK inhibitors, it is possible that if they're on a JAK inhibitor, that may help to decrease the likelihood of a severe inflammatory reaction. Again, we don't know this beyond a shadow of a doubt, but at least that is a possibility. Now, another piece that has come to light is this issue of an increased risk of blood clots and strokes in individuals that have been affected with this virus. We do not yet have proof that this has translated into an increased risk with MPN patients. However, we are concerned that MPN patients have a higher risk of developing blood clots and strokes just related to their disease. So we certainly have this as a concern, and we'll talk about what that means in terms of therapy at the moment. Now, should we adjust the dose of cytoreductive therapy, hydroxyurea, pegylated interferon, or even JAK inhibition in MPN patients, irrespective of COVID? And in short, we do not feel that that's the case. In short, we feel because of the risk of blood clots and bleeding, as well as because we feel that the risk of an MPN patient that are undertreated for their disease or probably have higher risk with COVID, it's best that people remain on their therapy and be adequately controlled. Now, if the dose of that therapy has been too high, meaning the white blood cell count is significantly lower than normal, we may slightly adjust the doses to try to get the white blood cell count closer into the normal range. We have encouraged maintaining good MPN disease control, and we certainly have encouraged for you to remain in close contact with your healthcare team, whether that be in person or increasingly also using electronic communication, electronic visits, which most centers and physicians now are able to offer. I've done dozens of these, interact with patients, both new patients and established patients. We talk about the blood counts, we talk about COVID, we talk about where they stand with their disease, and we keep in close contact during this dynamic situation. Now, should JAK inhibition be adjusted? Well, at first, we had a great concern, and I know of at least a couple of examples where people abruptly stopped their JAK inhibitors due to concerns about developing COVID-19. And in short, we would advise against that. Once a patient stable on a JAK inhibitor is probably much better off than a patient unstable who has come off a JAK inhibitor who needed one. So remaining on your JAK inhibitor, whether it be on a active clinical trial with momelotinib, with pacritinib, or if you are on commercially available therapy with either fedratinib or ruxolitinib, remaining on those, remaining well controlled is beneficial for you. If for whatever reason you had to come off JAK inhibition, then a appropriate taper would be in order. If a patient ends up developing COVID and is hospitalized and is on a JAK inhibitor, your hematologist clearly should be involved in the healthcare decision-making process, in that we would really want to avoid a non-hematologist abruptly stopping a JAK inhibitor during that time. That probably would not be advisable to rapidly stop a JAK inhibitor in the face of infection, particularly if it was more significant. Initially, at the beginning of this time, we had advised patients that needed a JAK inhibitor to wait on initiating JAK inhibition. And this is, again, at the beginning when we thought that the peak was going to come and go quickly, and we were going to have this only as a few weeks. Now that we're looking for a more extended period of time, I think that starting a JAK inhibitor, if it is necessary, should be done, but should be done with close observation. Now, again, at the beginning of this crisis, where we hoped that the period of risk was going to be short, we were trying to have just a holding pattern, try to treat people in the most basic way possible. I think now both the dynamics of at least a few months of a period before we have hopefully effective vaccines or things of that nature, really a stable phase. It's important that if you have a healthcare need, that it be addressed. So if you have an MPN, you should be receiving the therapy that you should be receiving normally, but with close observation. Now, if you develop the infection, should we adjust the MPN therapy? So one thing that is important is to watch for drug-drug interactions. And this is nothing that you as a patient has to monitor, but this is something very important for the healthcare team. Many of the antiviral therapies do tend to have drug-drug interactions and may require dose modification of your anti-MPN therapy. Additionally, rapidly stopping your MPN therapies. Let's say you're on hydroxyurea for controlling your counts. Abruptly stopping that might increase your counts and increase the risk of blood clots or bleeding. So again, that's why you need to stay in close contact with your hematologist during this period, both if stable, but also clearly if you become infected with the COVID virus, your hematologist as part of the team is an important part. Now, what about blood thinners? So individuals that are already on blood thinners, and by blood thinners, I'm referring to therapies like Coumadin or Eliquis or other oral blood thinners, different than, let's say, aspirin, which we would continue anyway. People on these therapies, we need to continue them without question. Individuals that become hospitalized, even in non-MPN settings, many of them are going on injectable blood thinners like low molecular weight heparin to try to protect against impact and effects. And likewise, with MPN patients, that is certainly a consideration. We've not called for universal starting of anticoagulation in all COVID-infected MPN patients, but it's at least a consideration to have with your physicians, but also monitoring the counts carefully, because on blood thinners, the risk of blood clots is less, but the risk of bleeding is heightened. So we need to navigate that path carefully. So I'll leave you with these take-home points. An uncontrolled MPN by undertreatment probably worsens the COVID risk. MPN patients overall are not severely immunocompromised, but some still likely have a higher risk than the general population. Maintaining patients on stable therapy, including JAK inhibition, is advised, and the potential of switching to low molecular weight heparin if infected is an important consideration. And with that, I'll hand it over to Anne, and Anne, you can decide whether we take questions now or have Aaron give his talk, and then we take some questions together.
I'm not sure if Aaron is with us yet, but I have a slew of questions. And
all right, well, why don't we start taking?
If you're able to take these now. And Ruben, I have several that are COVID related and several that are general questions. So how about if we do COVID first? And do we have, or is there a database that has begun to build MPN patients who have gotten the COVID virus and how they're responding to treatments and how they're doing? Is anyone compiling that database yet?
Yes, there's more than one, but yes, everyone is trying to track this information for us to learn from it to the best degree that we can.
Okay, and can you explain the various immune suppressions, for example, with ruxolitinib that MPN patients might have, and how they might relate to one's health on the risk with coronavirus? You kind of answered that, but
well, let me try to. Immunosuppression is not a yes/no. It really is a spectrum. So at the far extreme of that spectrum are individuals whose white blood cell count is incredibly low, individuals with acute leukemia, individuals undergoing bone marrow transplantation. These are individuals at profound risk of multiple severe types of infections. I would say MPN patients are somewhere on the low end of that continuum. But JAK inhibitors and ruxolitinib, of which we have the most amount of time following the drug, does increase the risk of infections somewhat. We don't think dramatically, but clearly there are some that it has. So one, some viral infections, so increased risks of the herpes shingles virus reactivation, probably an increased risk of respiratory infections, increased risk of atypical infections reactivating like TB or hepatitis B, and some very rare viral infections. So all of that said, the majority of patients that have taken ruxolitinib have not developed an infection, but there's a little bit of an increased risk with some viruses. We do not have any specific data yet that says that ruxolitinib, fedratinib, pacritinib, momelotinib, that any of them specifically increase the risk of COVID.
So someone asked, and I have the same question because I think it gets very confusing about testing. So, if I'm symptomatic and I get tested, and of course it shows that, yes, I do have the virus, and depending on the extent of which my symptoms are, if I'm very desperate for hospitalization or whatever, but let's say I am asymptomatic, I test negative, and 2 days later I'm very sick. How do you feel about testing?
So one, as we talk about testing, there's two different types of tests. There's the test to see whether you have the active infection now, and the other tests that have not been extended to a great degree, which are the antibody testing, meaning you have had it in the past and now have antibodies. So let's start with that one first. What is hoped for the antibody testing is, does that identify people that, when did you have it, which is of an interest kind of from an epidemiological standpoint, or maybe out of a personal curiosity standpoint. But the more important question, are you protected or are you immune either now or for a period of time, it may not be permanent, against getting the infection again? There is great hopes that individuals do become protected. The rate of reinfection, as it has been studied in Asia, looks very, very low. So they've looked at this very scientifically in South Korea. They're looking at it in New York. They're looking at it in other areas. We are hopeful that people that have recovered at least can't get it again, at least for a period of time. Whether that's a year, is that 5 years? Nobody knows yet at this point. Likewise, if you have a positive antibody test, are you protected? Again, I think that we're only learning. Part of the challenge in developing that particular test is that being certain that you have antibodies against this specific virus, because the first one of these tests that were coming out had a lot of false positives for people that had immunity or antibodies against the cold virus and not against this specific virus. So that really they wanted to avoid telling people they were protected, and they were not. Now, in terms of the test for the virus itself, usually it's in the setting of helping someone make a decision, meaning you have another healthcare issue and you want to know whether you're infected. You can now likely probably get tested, at your medical center. Now, the availability of testing, in the beginning when it was very limited as to who could get tested. Now in San Antonio, there's dozens of places you can just drive up to. You can get tested if you wish. Now, does it give you any useful information? It depends. So here we are testing asymptomatic people who are going to start chemotherapy, start radiation, going to be hospitalized for whatever reason, undergoing surgery, having a procedure. So we're doing that piece, and anyone who might undergo anything that might be further immunocompromising or expose the rest of the healthcare team, we know whether they have it or not, and whether we defer. So if somebody's getting chemotherapy, we may wait for 2 weeks if they are asymptomatic, positive, or things of that nature. If you have it and you're positive, your healthcare team should know. Now, they may well likely interact with you with an e-visit. Being asymptomatic positive does not mean you rush in to see your doctor, and it certainly does not mean you rush into the emergency room, unless if you feel sick. If you feel very sick during this time, if you have a shortness of breath, whether it's COVID or not, you always should be seeking emergency medical care. But if you're asymptomatic positive, good to connect with your doctor, either your primary care doctor or your hematologist, in terms of what the next step is.
Okay, and now moving on to more general questions about MPNs. We've had this discussion, and I've heard conversations or presentations by you and others about allele burden. Can you explain, about just explain the allele burden, if it's higher, is this person suffering more? And, those types of questions are coming in.
Sure. So what the allele burden means, if we looked at 100 white blood cells from an individual, what percentage of those white blood cells have the mutation question? So if we're talking about JAK2 V617F, is that 50% of the cells, 75%, 25%? At the current time, we don't know whether the allele burden is a overly helpful marker of the disease. In other cousin diseases to MPNs, in chronic myeloid leukemia, the allele burden has been a helpful barometer, but it's a very different disease. So in MPN patients, we do know that patients that have a higher allele burden might have more P-Vera-like features. So if you have ET and you have a higher allele burden, you probably have a higher red count, you may have a higher white count, you may have greater likelihood of symptoms or itching. But we have not necessarily found that having a higher allele burden is predictive of the disease progressing, being more severe, or being more threatening in any way. So I think it's a marker we continue. I think that we need a great MPN marker, but I don't think allele burden is likely it. I think that there are potentially other barometers of the disease that we have not yet discovered that might be more predictive for us. Again, for most MPN patients, the thing that we are the most concerned with is the risk of progression. Right. And the allele burden is not really the predictor of that. I think once we find truly why MPN patients progress, we might be able to track that better, track the response to that better in response to therapies, whether that be interferon or JAK inhibition or others, and be able to adjust accordingly.
Okay. How about one more question, Dr. Meza? I know you want to hear Dr. Gerds and maybe have a little discussion about that, about what he's presenting, but at what age does the bone marrow quit producing platelets? That's a good question.
It's a good question, but the bone marrow keeps producing platelets right through the entirety of life. The normal blood count in someone who is 100 years of age is the same normal blood count as someone who is 25 years of age. So the blood counts do not adjust based on age. So although there are times that people do not do a workup for abnormal blood counts in people that are significantly elderly, they should not decrease. So if they do, it is a sign that some medical problem has occurred that we need to be mindful of.
Okay, too bad other things don't stay the same as we age. So with that, thank you so very much. I know your work has been extensive these days, even more than you probably anticipated. We will move on and introduce Aaron, and any questions that I have here that were not answered yet, we will get answers via email to you. And thank you, Ruben. I really appreciate it. We'll be in touch.
Great. Thank you, Ann.
So at this time, I'd like to introduce Dr. Aaron Gerds at the Cleveland Clinic. He has been also a frequent speaker with us, and he is going to talk about the other side of COVID and some other things that MPN patients should be aware of. And with that, thank you, Dr. Gerds, for being with us. I know it's a busy time for everybody.
Well, thank you for inviting me. I'm absolutely thrilled to be here to share with you some things that have gone on here locally and some thoughts about how the MPN world has been affected, particularly clinical trials in this current era that we're in with the pandemic. And I know Ruben covered a lot of ground about MPNs and COVID-19, just COVID-19 in general, and I'm hoping to take that to the next step further of how COVID-19 and this pandemic has affected conducting clinical trials and perhaps some opportunities going forward. All right, so now I want to show my slides. I apologize. I don't use GoToMeeting a whole heck of a lot. All right, is it showing my slides now?
Not yet. Oh,
All
here they are.
right,
Yeah.
there we are. Yay,
Great,
it works. All right. Deep breath with the technology. All right, so clinical trials in the COVID-19 era. So I do want to make a disclaimer here first. If you're neckwear adverse, I do apologize, but we are going to proceed forward nonetheless. All right, move on to the next. All right, so we're going to talk about very briefly new treatments being developed for MPNs and kind of to put clinical trial development in a framework. There are a lot of clinical trials ongoing even in this era to improve patients' lives with MPNs, improve how they feel, improve delay progression, improve the way we take care of these patients with these diseases. And so I think that's really important to remind ourselves of. But obviously things are different nowadays, and clinical trials look a little bit different during the pandemic than they did in times prior to the pandemic. And then there's actually a lot of crosstalk between MPNs and COVID-19 research, namely in the realm of cytokines and cytokine storm. So I want to briefly touch upon that as well. So starting just with new treatments being developed for MPNs. Now, this is just a quick taste. You know, like when you go to the ice cream shop and they give you that tiny little spoon of just a little bit of ice cream, you get 3 samples and then you got to kind of pick your choice. This is just a taste, and this is not by any means an exhaustive list, but more of a framework of how to think about the different trials that are ongoing for MPNs and organize them in our minds. That way we can kind of get a grasp of the whole picture. And the way I like to think about it is where we are now, things that are just ahead, medications that and treatments that are really close to the finish line and can impact the care of patients with MPNs in the very near future. Things that are further down the road, that trials that are ongoing or maybe a little early phase, but maybe have a bigger impact than some of the stuff that's just around the head, just ahead, these things that are kind of quick wins versus so that's going to be a little bit more long game, and certainly on the horizon. How are we thinking about the clinical trials after that and the treatments after that? How can we really delay progression, cure without transplant, colon eradication, these really big, lofty goals, and for treatments that are, again, a little bit further down on the horizon. So where are we now? Many of you may be familiar with the treatments that are commercially available at this time, including hydroxyurea, ruxolitinib, fedratinib, interferons, the erythropoiesis stimulating agents, which are specifically aimed to improve red cell counts and anemia in patients who have that anemia. The IMiDs, such as lenalidomide and thalidomide, these immune modulators, and danazol, which also aims to improve anemia, as well as some chemotherapies such as azacitidine and decitabine, as well as allogeneic hematopoietic transplant, which is currently the only therapy that can potentially cure people with an MPN. So there are some things that are just ahead, kind of, I don't know if I like the term quick win, but things that are just within reach that could become commercially available in the very near future, again, to improve patients' lives with MPNs. The one drug up here on the top, something called luspatercept. It's a medication, again, to improve anemia in patients who have anemia, improve red cell counts in patients who have anemia as a result of myelofibrosis. It's already approved for myelodysplastic syndromes as well as thalassemia, but we are working towards developing data in MPNs to hopefully help expand the use of this medication to patients with MPNs more readily. Other drugs like momelotinib and pacritinib, these kind of newer JAK inhibitors, are being developed and are moving into phase 3 trials. A little bit further down the road, you can see the focus of the treatments of a lot of these, the focus of treatment for a lot of these agents is to get under the disease, not maybe just focusing on symptoms and spleen size, but really getting in there and, say, killing MPN cells really modifying the disease, if you will. So drugs like the PI3 delta kinase inhibitors or the SMAC mimetics, which actually their whole goal is to increase apoptosis or really kill off the myelofibrosis cells or the MPN cells within the patient's bone marrow. Or drugs like AVID 200 or PRM151, which have a goal of reversing the scar tissue in the bone marrow. So these are really getting at the disease itself and what we call disease modification. How can we reverse the processes that have been going on over time within a patient's bone marrow? Again, to not only improve their symptoms and say spleen size and control their counts, but really affect the course of disease significantly. And again, on the horizon, these things are bigger picture stuff. And a lot of these treatments are still very, very early on. These are things that are even before the clinic and are worked on in people's research labs and in kind of in petri dishes and animal models right now. Really kind of lofty ideas of where we can take the science that we have right now about MPNs and how they develop over time and really get in there and clean out these diseases. And at one of our MPN meetings not too long ago, one of the speakers, Dr. Mascarenas, got up and said, you know, really the goal here with a lot of these projects are to cure patients with MPNs without having to use a transplant. Again, transplant is a great modality to cure people with this disease and the set of diseases, but really what would be wonderful is if we can use the knowledge that we have of the biology and really get after it, modify the disease to the point where patients don't necessarily need to do transplantation, but yet have long-term maintenance-free survival. So this is the landscape and kind of, again, a context and a framework to organize the ongoing clinical trials in MPNs. But of course, things are a little bit different right now. We are currently dealing with this pandemic, which has changed how we think about clinical trials. And just a couple of things that come to mind about clinical trials in this area is, one is, of course, safety. We want to make sure our patients are safe. We want to make sure that people taking care of our patients are safe. And that has changed what we do in the clinic. There are now screening procedures at many institutions where you walk in the door, you're getting your temperature checked, they're asking you about symptoms. We're also trying to, quote unquote, decrease the number of touches, how many times a patient has to come in to get a blood draw or a CAT scan or see their doctor or nurse practitioner or their care team. And so we're really trying to think about how can we keep patients and caregivers safe. That's the number one thing. When thinking about clinical trials and what comes after, once you complete a clinical trial, you want to get the results and you want to interpret them. Many trials have been put on hold or have changed their processes, and that may affect how we can interpret the results. Trial accruals have slowed nationwide, and that may affect the way we can understand the results, particularly if patients do stop their participation in clinical trials, it may be difficult to get long-term safety data. Also, with the less touches and less lab draws and some of these modifications that trials have undergone in this area, we may have less correlative studies. So the science that feeds the clinical trials and our understanding of these medications and how they work, and where we get ideas for the next line of treatments and how we might attack this disease in a different way, may be compromised by having less correlative studies where we get the blood draws or the bone marrow biopsies to analyze them while patients are undergoing the clinical trial. The funding sources have also been affected. So a lot of federal funding is being redirected to COVID-19 research directly, and appropriately so. This is a big issue and we need to tackle it head on, but that may be perhaps a slowdown of focusing on other diseases. Philanthropy has been a big source of supporting research, not only for MPNs, but a lot of diseases, a lot of cancers. And there really has been a downturn in the market as well as in philanthropic donations. And so that may change how clinical research is funded. I also mentioned earlier the feeder for a lot of our ideas and how we take treatments from ideas into the clinic is through basic science and translational science, where scientists we partner with get the samples, they do the research, they unlock the mysteries of this disease. It's so critical for that part of this process to feed into the clinical trials to get these treatments to the clinic for our patients. And maybe basic science and translational labs have been shut down or are operating under minimal staff. And setbacks from this could be up to 6 to 12 months, where the research has basically been put on pause. And so that may affect how we take the science that we're trying to learn and translate it to patient care. So what I can speak a little bit more locally to how we here at the Comprehensive Cancer Center have been thinking about COVID-19 and clinical trials. Again, our utmost and primary concern is the safety of our patients, as well as our research staff. And we took a number of steps here at our institution to try to really modify things. We went through trial by trial on a case-by-case basis, and trials with moderate and low potential of benefits. We discussed whether or not we should close them, or at least suspend them for now, or flip them over to a virtual type of trial. So, for example, a trial where we may be following patients for quality of life or symptom burden. A lot of times we can do that virtually through virtual visits or telephone calls. A lot of studies that were just serial samples where patients would come in and donate blood for research, many of those have been suspended or even closed. We have taken a lot of effort to try to keep trial participants informed. We've generated letters of how each individual trial has changed and what we are doing to protect patients and keeping their safety in mind all the time throughout this. So we're generating letters as well as phone calls to keep folks informed about what's going on and what things are changing. We've shared best practices within our own institutions, as well as other cancer centers across the country. And there's really been a large sharing that has occurred. I know Dr. Meza pointed out some of the things that ASH and the American Society of Hematology have been doing to share information about how to take care of patients in this era. We've been doing the same with clinical trials, how we can best conduct clinical research, keeping our patients safe, our caregivers safe, but yet getting meaningful results during these times. And these have been a lot of parties have been involved with these decisions and these discussions, including the people who fund the studies or make the medications that we're studying. Certainly the FDA and the institutional review boards have all been part of these decisions, and they're really multilateral in nature. And looking across our clinical trial portfolio within our Comprehensive Cancer Center, roughly 10% of clinical research studies have been put on hold. The proportion of the studies that are not interventional, meaning patients aren't getting treatments with those clinical trials, that's the larger number there, but a few clinical trials where patients actually get treatment have been put on hold as well. But about 10% of clinical trials have been put on hold. So we are trying to continue to conduct this vital research and provide the access to the clinical trials during these times. For those of you in the business world, you may think of a SWOT analysis. And so the important thing always at the end of these situations, when you've gone through these processes, is, well, what are the opportunities here? What can we learn from this? What can we take forward over time, even when this is all past us and we're returning to the new normal, or what will be the new normal, and we're emerging from this? What can we do better in the future? And one of the main things is, maybe we can do a better job designing clinical trials. Do we really need to have this many visits? Do we really need to collect this certain data or these correlatives? How can we make clinical research better? And this may be an opportunity to help us in the future do a better job at designing clinical trials. So I mentioned earlier too that the pipeline has been affected. The science that is being conducted in research labs has been put on pause. And so all those folks who are at home, staying at home like they should be, and not in their research labs, a lot of them are at home writing grants, writing the next and thinking about the next wave of research. So when labs do reopen over the next several months, I can anticipate a huge wave of research being done. And everyone is very excited and being very thoughtful about their next projects and everybody wants to get back in the lab and get working. So I expect there could very well be a wonderful and amazing burst of scientific activity in the MPN space in the coming months. Also, collecting data on how patients with MPNs do in the COVID era, with an infection, without infections, can really maybe help us understand better how infections, particularly viral infections, affect patients with MPNs. And that's something else we may be able to learn with efforts like the ASH, or American Society of Hematology, the ASH Research Collaborative, which I'll mention here in a moment. So this is a registry. The ASH Research Collaborative is a large group of institutions from around the world that are pulling together to do research in blood diseases, not just MPNs, but blood diseases in general. There are efforts in sickle cell disease with clinical trials. There are large registries again and retrospective databases that are being done through the Research Collaborative. But one thing that is launched recently is a registry specific for patients with blood diseases who have COVID-19 to try to better understand the complications from the disease and what's going on with these patients, and so we can all share information and share it real time. And so anyone can go to the ASH Research Collaborative website and pull up this data. And so this was as of May 21st when I pulled these figures from the registry. 141 cases have been reported so far. 15 of those patients had MPNs. On the right, you'll see a map of the world, and this is where the patients that have been put in the registry are coming from, mostly from North America, but actually it's a global effort. Specifically on the left-hand side there, you can see the breakdown of the MPN patients, roughly 50-50 mix of the MPN patients. A mix of severity of disease on the top there between mild, moderate, and severe. And a broad range of ages there, with the 70-79 age group kind of sticking out a little bit, but patients as young as in their 40s or as advanced in age as the 80s have been reported so far. Again, these numbers are very, very small, but the registry is collecting information about the treatments that these patients are receiving for their disease. And so again, this is specifically the MPN patients that have been reported so far, and patients are often receiving treatment at the time of their COVID-19 diagnosis. So understanding the interaction of the COVID-19 virus along with their treatments is going to be something that we can learn from this registry going forward. There's also a lot of crosstalk between MPNs and COVID-19 research, and that crosstalk is really centered around cytokines. So as Dr. Meza mentioned, COVID-19 causes this cytokine storm, or this severe inflammatory reaction. Sometimes we also use the term sepsis, where the immune system is really, really revved up, producing all these cytokines, and a lot of the consequences of the disease are a result of that. Just so happens that a lot of MPN treatments focus on dampening cytokine storms or cytokine reactions or cytokine levels, if you will. And so it's only natural that treatments for MPNs may be applied to the care of patients with COVID-19. And in fact, there are a number of studies that I have launched, and even more that are planned for the treatment of patients with COVID-19. And there are 9 studies to date that are far enough where they have registration processes done for ruxolitinib, and there are 2 studies that are ongoing with interferons, and other studies have been planned for medications like pacritinib. This is the global coronavirus clinical trial tracker. Again, this is available on the internet. I pulled this up on the 21st, so that's data as of then, listing all the clinical trials that are being conducted. And you can go to the bottom of here and click on the actual links, and it takes you to the clinicaltrials.gov website where the more information about the individual trials have been posted. But this represents This map here represents the location of the 11 trials with either ruxolitinib or interferons that are ongoing. So again, here's a couple of screenshots and a little bit more information about ruxolitinib. Again, ruxolitinib, the thought is it can block the cytokine storm in patients with really bad lung disease from COVID-19. A lot of the inflammation and cytokines are very, very high in these patients, and by tamping that with ruxolitinib, could potentially bring patients through that period quicker with less complications. And again, there are other trials being developed using other JAK inhibitors such as pacritinib. Interferons, the way the thought is it works in infections a little bit different and a little bit more towards the disease itself. There is data with this other coronavirus that happened a few years ago, the SARS virus, if you remember that. There was information and clinical trials, information developed from clinical trials using interferon in SARS. And really the way that we think interferons work is it's a broad spectrum antiviral drug because they activate these things called phagocytes. And you can think of them as almost like immune cells that work like Pac-Man. And they are at the site where there's inflammation, where the virus is, and just kind of gobble up cells that have been infected by the virus and eliminates them from the body. So kind of a broad spectrum antiviral is the way we think about interferons in this setting. And so a number of clinical trials have been developed. Two, again, I have launched with interferons, some being subcutaneous, others being inhaled, in fact. Inhaled interferons where the interferon goes directly to the lungs where they can help with the COVID infection. So, so far we've covered new treatments being developed for MPNs. And really, again, I wanted to put it in a framework for you. We could be here all day long talking about all the wonderful clinical trials that are ongoing to improve the lives of patients with MPNs, but really the way I like to think about it are treatments that are already available, treatments that are really close to being out there to helping people, you know, drugs that are very close to commercialization, drugs that are almost ready for prime time. Then things that are a little bit further down the road, more middle of the road, kind of more advanced clinical trials like phase 2 studies, where those studies are really kind of getting at unrooting the disease and disease modification. And then lastly, trials that are on the horizon, things that we can think about where it's really going to reverse the course of disease. Clinical trials have changed in these times, and we're really thinking about how can we do clinical trials incredibly safely, keep our caregivers safe, as well as get meaningful results during these times. And so clinical trials have been modified, but many centers are really committed to offering clinical trials and continuing to offer new and innovative treatments and pushing patient care forward. MPNs don't stop happening because of COVID. People are still going to get PV, ET, and myelofibrosis, and we still need to be there to take care of patients and advance the science of taking care of these patients. So I think that's a really important point. We're trying to take advantage of what we know of MPNs and cytokines in general and how these drugs that we have used for years work, and actually funneling that back and cycling it back to direct COVID-19 research as well. And maybe we could potentially repurpose some of these medications to help patients who have COVID-19, even though they don't have an MPN, but really help in the greater need at this time. So I'd like to thank you for your attention. And I think at this point, we will move to the Q&A.
Hi, Aaron, can you hear me?
Yes, I can.
I have a ton of questions.
Oh boy,
Okay.
Good. I like questions.
All right. One is if you suspect you have COVID, can you still join the ASH registry?
Yeah. So yes. So the ASH registry is for any patient with a blood disorder who has COVID-19. So your doctor could enter your data into the registry.
Okay. My 18-year-old son had ET and is JAK2 positive. As his mother, I worry about the clotting aspect of COVID and ET. Can you address that, please?
Absolutely. And I think Dr. Meza touched on this a little bit. It's a challenge because we don't know exactly how much it increases the risk. You know, really the way to think about clot risk, at least I do it very simplistically, it's all additive. And so if you have underlying factors that increase your risk for clots, and then you get sick from, say, COVID-19, or quite frankly, any infection, that can increase your risk for clots. And so that adds up. So that is something that can be very worrisome. With the clots in COVID-19, it really appears to be the patients who are very sick. Those are the ones who are most likely to get clots. So I think for the average person who isn't sick, you know, the risk is not any different than it would be in any other different times. But so that's where the recommendations are coming from right now, that patients who are on some sort of antiplatelet therapy or a blood thinner, if you will, continue on it for now. You know, in people who do get COVID-19 infection, and to follow the guidelines for anticoagulation there that have been established.
Okay, Aaron, when oh, he just disappeared.
Oh, no.
When you say down the road, you know,
Yeah.
I think Ruben used that term, but how many years are you estimating until we can all live a normal life? This is just from a patient.
You know, that's the billion-dollar question. I think my sense is it's going to be a very gradual process, and down the road is going to be contingent on a couple of things. People in this country on a whole have done a wonderful job staying at home, taking the precautions, and quote unquote flattening the curve, if you will. And you know, one of the consequences of flattening the curve is that the curve gets wider. It's like you're literally smushing, and so things take longer to get through this. So that, I think, affects it greatly. The other thing that will affect quote unquote getting back to normal will be developing a vaccine. That's a pretty critical part of that. And if we can protect the population without having the virus have to go through the whole population, that's really gonna shorten the time. So to me, if truly a vaccine can be developed in the next 12 to 18 months, I think that could really turn the tide and get us quote unquote back to normal much sooner. Otherwise, we have to wait for enough of this to go through the population where we can safely reopen more and more things.
Ruben, if you're still there, would you like to respond to this as well?
So first, Aaron, wonderful talk. You know, I do think that, you know, the rate of progress is going to be very rapid. You know, how complete it will be, you know, and how soon, I think, is difficult to predict. I do think that, you know, in the future, you know, we are going to have greater risks of other infections coming through. And I think as a society, we're probably going to be more poised to that. You know, our infection experts do predict that, you know, this virus mutated from the other one, but that it's continuing to mutate. And you know, the process of us being more mindful of public risk of infection, I think will be higher, without question. I do think that the likelihood of some sort of vaccination strategy probably will start very soon for this. Now, is that September? Is that January? I don't know, but I know that the pressure is beyond comprehension, and the number of candidates, you know, being tested in parallel is dramatic. You know, so I could certainly see a situation where we all get a vaccine and it's okay. And then we get another vaccine 6 months later that's better. So I mean, I think anything is possible, but I think without question, everyone is thinking global. You know, how do you find a vaccine? How do you make 6 billion doses? I mean, people are really thinking in those kind of terms, as well as, there's a standing order the U.S. has for the first 300 million doses of a vaccine. So I mean, people are thinking on that sort of scale, but how effective that first vaccine is, how long it takes for people to get it. Again, it's going to be a process, but I see it happening as soon as it is possible.
Yeah, I mean, if you look back at history, so you look at the polio vaccination, right? So the first vaccine was incredible, but it wasn't perfect. And then, years later, there was a second vaccine that definitely moved that effort. And same with shingles. We're all familiar with shingles. So there was the Zostavax, which was pretty good and kind of went through. And then all of a sudden there was Shingrix, which is better. And so, but that was years. And it's just incredible to see the effort behind this that is shortening things that take half a decade down to months, if you will, or even just a couple of years even. So it's pretty amazing what's going on right now. But to me, I agree that that's going to be the linchpin of how we get back to a more normal life, but also agree that we're going to be I think we're going to be incredibly mindful of these things going forward. And I think a lot of our habits are going to change and change permanently. So were there other questions,
Okay,
Ann?
gentlemen, can you hear me?
Yep.
Sorry.
Yes.
Can you hear me? Okay. So the next question is one that I think weighs on all of us, but particularly on MPN patients. And that is, when allowed to go back to work, or currently we are allowed to travel, some states are allowing, more travel. Is there any particular, thing that an MPN patient should do to ensure their safety, or is it just the stuff that the general population is doing? And a caveat to that is, are cloth masks safe enough?
All right.
Do you want to go first, Aaron?
Sure. Well, definitely everyone should be doing their part for themselves and for their community about, again, for now, staying home, but then also, washing your hands regularly, wearing the masks if you have them available. I think all those are very important, and just as important for MPN patients. In addition to avoiding sick contacts is an incredibly important part of all that. Anything extra? I think it's really on a case-by-case basis. So if you're an MPN patient who has, say, ET, and you're otherwise young and healthy, and you don't have a lot of other risks, again, risks always tend to stack up and are very additive. I think, the general precautions are what you should be doing. If on the other extreme, you're, say, a myelofibrosis patient who just had an allogeneic transplant, that's obviously the other end, and your precautions need to be much more stringent, for sure, as the risk, not that you get the infection, but that you have a complication or a rough course with the infection is much, much greater.
Yeah, I would say, if you're really following what they're recommending for the general public to do, that probably is good enough for most. It's just actually doing what you're being recommended to do is the key part.
Yeah, and being diligent about it. Myself too, like, I'm probably, I have to keep reminding myself to simply, like the washing the hands part. When this first hit, I was super diligent, and then I had a week where I kind of slid a bit, and now I'm back on it. It's kind of keeping with it and keeping on top of it is key.
Okay, can you gentlemen hear me? If chemo meds are needed as treatment, will these meds affect other cell lines? This is not a COVID question.
Yeah, often chemotherapy affects a number of cell lines. Very few of our therapies we have today are incredibly selective, where it just goes in and gets one precise cell line or one particular variety of cell. They often have a predilection for going after the MPN cell versus a normal cell, or one cell line versus another, but it's really difficult with the current medications we have right now, both commercially available and ones that are in more advanced clinical trials, to be that precise. So whenever you look at, say, the package insert for, say, ruxolitinib or fedratinib or a lot of these other medications, yeah, it mentions lowering the blood counts in general as a potential side effect.
Ruben, did you have anything to add to that?
No, I think that's good.
Okay. And then in the case of biological and hematological remission, do symptoms cease? And what is the best drug available for remission, and any other factors besides drugs involved in remission?
Well, perhaps I'll jump in. The word remission, is a word that means that there's no absence of disease, but we don't necessarily think that the disease is cured. And disease by disease, that may mean something different in one disease versus another. What that means in breast cancer might be different than in acute leukemia. In MPNs, we, for the most part, do not, have a remission state, short of people that have been successfully treated with allogeneic transplant. Now, whether that will evolve, I think is unclear. There certainly has been discussion or controversy regarding whether remission is something that's been seen in some patients who've been treated with interferon. Is the disease really gone? Is it dormant? Can't we measure it? It's been in a very small subset of individuals. Now, that said, there's a lot of individuals that are not in a remission, but they are on a plateau, meaning that the disease is stable, it's controlled, they overall feel pretty well, their blood counts are overall pretty well controlled, and there's no evidence of the disease progressing. So I think in the future, although if we get to the point where we can see no evidence of disease, that would be great. At the current time, we don't want the perfect to be the enemy of the good. So in a lot of patients, if we can achieve a plateau, that is a very favorable state. But in research, without question, we hope to be able to get to a remission state. But for the most part, that is not something we're achieving at the moment.
Okay, and this is a question from a young woman who is pregnant or is asking about pregnancy with essential thrombocythemia. Is it safe to be pregnant? What are some precautions she's going to need to do? Aaron, did you?
Sure, I'd be happy to jump in. There are a couple of things that we could recommend, and actually, you could always inform your caregivers as well as yourself. There are guidelines that are available to review for patients who have MPNs, and they're posted on the NCCN website, and do actually address pregnancy and MPNs as well, which is fantastic. That resource is available for patients and their doctors. In general, there are a few things that I would recommend for a patient with ET who is pregnant. Certainly, we want to avoid anything that would harm the baby, harm the fetus. So things like hydroxyurea are obviously can be harmful. We would want to not use those. One of the other kind of key factors is the clot risk. So not only clots in the mother's venous system, but clots in the placenta that could affect the blood flow to the developing baby. I think getting coordination between your hematologist or the person taking care of your essential thrombocythemia and high-risk obstetrician is important, and having that dialogue back and forth, because these people are both part of your team and really need to work together to come up with a plan for you as an individual. Lots of guidelines do recommend aspirin throughout parts of the pregnancy to help reduce the risk of blood clots in the mother and the placenta, as well as either pre-delivery or postpartum anticoagulants or blood thinners, kind of depending on the situation. So a lot of these decisions are made on a case-by-case basis, but there are some guidelines on how to think about and frame that for an individual patient, again, within the context of a coordinated care team, including both your ET doctor as well as a high-risk obstetrician.
And to add to that, we will be hosting our 7th annual MPN, Women in MPN program in September online. And they will address, very specific pregnancy, issues, and those conversations will take place just in case. All right, how about 2 more questions and then we'll wrap it up. One is, will it be safe for MPN patients to take a COVID-19 vaccine if the government mandates that? Do we even know if that would be safe?
I think it's just premature to know.
Right.
But overall, with vaccines, there's the vast majority of vaccines, use part of a virus to create an immune response, as opposed to the whole virus that can infect someone. And there's a couple examples of the type that are live virus that we can shed. I would tend to think in terms of how all this process is ongoing, that it's highly likely to be a non-infecting type vaccine that will move forward for a range of reasons, including, decreasing the risk of secondary infection and things of that nature. So I think it's highly unlikely there's going to be a vaccine that immunocompromised patients wouldn't be able to take, because it semi-defeats the purpose of developing the vaccine.
Do you want to add anything to that, Aaron? Or is that...
No, I think it's hard to top that explanation. I completely agree. Yeah, in all likelihood, it will be a dead virus, or virus chunks, if you will. It's pretty unlikely to be a live vaccine, and so it's most dead virus or chunks, if you will, of viruses. It's safe for patients because it can't cause an infection because it's just proteins or polysaccharides. So it should be ideally safe for people who need it the most.
So the last question, getting back to, I believe, Ruben, you mentioned this as well, progression. So, we're hearing more and more from ET patients who are, their first diagnosis is essential thrombocythemia. And as a younger patient with ET, my concern is progressing to MF. Is that the natural progression?
So, Aaron, you want to start us off?
Sure, absolutely. So it's not the natural progression for everyone. Not every patient who is diagnosed with ET will progress to myelofibrosis or even to accelerated blast phase disease, acute leukemia, if you will. I think we, over time, are better understanding which patients may and which patients may not. We look at second or third mutations that may be involved, or fibrosis or other markers that may help put into different categories people who are more likely to progress than the others. But certainly not every patient will ultimately progress. I think it's important to try to understand the risks that may make a patient higher or lower risk, and have a discussion around that, and certainly avoid things that may make progression more likely. But there's not every patient will progress, but I think close monitoring is a key component of managing that risk going forward.
Yeah, I think that's a great explanation. I do think that is the natural progression of the disease, but certainly it's a progression that is still the minority of patients that experience that. So the majority of patients with ET or PV will not progress to myelofibrosis. The factors that clearly weigh on that, the biggest of them is really time. Years and years of the disease, 10 years or more, is clearly a factor in that. Without question, we're learning more about the drivers as it relates to genetics and others. But again, it's far from a guarantee. The key is to be trying to take good care of your health, living your life the best way that you can. And meanwhile, Aaron and I and our colleagues will continue to work hard with all of our scientific colleagues, our colleagues in the MPN world, and our partners in the pharmaceutical industry to hopefully develop therapies that will stop that progression from ever happening.
Well, gentlemen, I can't thank you enough. I'm going to put, well, I was going to put myself on the screen. I can't. So that's okay. Oh, there I am. Okay. So I just want to thank you both so very, very much. I greatly appreciate your time and efforts. And I think you helped a lot of patients today and caregivers that were on this webinar. And I thank you for your patience. It's the first time we've done this to all of you who are joining us. We will be doing this subsequent webinars as we just determine when it will be safe to meet in person. This is our best alternative. And any questions that we've missed, we will send to the two physicians here and try to get answers for you. I'd like to thank very much our sponsors. We would not be able to do this without them. And again, I greatly appreciate your time and efforts. And to all of you, please be safe. If you need an emergency card, let us know. We've sent out hundreds over the last couple of months, and be safe and let us know if you need us. Thank you very much.
All right.
Bye-bye.
Thanks, everyone.
Take care.
Bye.